Now I'm going to move on to Kevin Slawin, who's the CEO of Senotherapeutics. We would call you guys a level 7 and potentially a level 8 company, Kevin. And of course, you've recently rebranded and you're kind of emerging from stealth. So you're focusing on the xenotherapeutic hypothesis of aging. And that's a terrifying amount of slides to get through in your time. But I'll hand over to you and good luck.
Uh, Thankfully, most of those are in the appendix.
That's great. Over to you,
Uh,
Kevin.
can you hear me, Phil? Okay.
Yep, we can. Yep, all good.
Okay, great. I appreciate the opportunity, Phil and Master Investor, to present Eos SENOLYTIX, our xenotherapeutic company that's goal is to improve health span and increase lifespan. Just getting through the usual legalese. So we're working in geroscience, which is the study of mechanisms that make aging a major risk factor for common chronic conditions and diseases of aging. And geroscience is really at the intersection between aging, disease, health span, and lifespan. The primary goal of geroscience is to extend the healthy years of life by targeting underlying biological process of aging. And there was a very nice a paper recently on the goals of geroscience that you might want to access. Um, Now, our company is focused on something a little more straightforward than perhaps Yamanaka factors, which is fascinating. We're working on what's called the xenotherapeutic hypothesis, which says that senescent cells, also known to the layperson as zombie cells, which can be identified in tissues by either high beta gal expression, which is blue staining with X-gal, or other immunohistochemical markers like P16 or P21, that they accumulate in various organs with age, and that these senescent cells secrete a pro-inflammatory set of factors called the senescence-associated secretory phenotype, or SASP, and that these cells and the SASP then leads to a process called inflammaging, or an inflammatory process in the body that's in no way linked to any kind of infectious process. It's just inflammation associated with and causing aging. Now, in this hypothesis, the accumulation of senescent cells as we age is the cause of many aging-related problems, if not most. So, if you go through all the major organs of the body and the brain, you get cognitive decline, Alzheimer's, CTE, the heart, kidneys, etc. We see all these aging-related diseases that accumulate with age that are directly associated with the accumulation of senescent cells in the body. And there's a very extensive proof of concept set of studies in the peer-reviewed literature that clearly shows that if you can eliminate senescent cells, selectively eliminate them from the body, you can reverse all of these aging-related diseases. And in fact, if you're interested in seeing this body of literature, you can see in the bottom right there, I've curated them on our website in the media center there, and you can click on any organ and see what I'm referring to. But in the brain, clear studies show that if you eliminate senescent cells, you can see cognitive and memory improvement. In the heart, kidneys, you have a reduction in glomerulosclerosis, etc. And so I think the proof of concept here is quite extensive and very solid. Not only can you reverse these particular aging-related diseases, but there's also clear evidence of both the extension of a healthy lifespan or health span and even lifespan, and a reduction in death from cancer. In simple terms, how would I explain this process to the layperson? Well, let's look at it like a drain. During our youthful years, we don't have a lot of senescent cells in our body, and the immune system is quite good at clearing them, so there aren't that many present. Your organs function smoothly, like a free-flowing drain. As you get older, the immune system's rate of senescent cell clearance is mostly keeping up with the rate of formation, so there's still not that many around, and the organs continue to function relatively smoothly. But as we continue to age, senescent cells will continue to accumulate, the immune system becomes less efficient at removing them, and the inflammation caused by the senescent cell itself causes immune system impairment, cellular stresses associated with aging, increase the rate of formation of these senescent cells, and this leads to their progressive accumulation. And now you start to see organs beginning to malfunction due to this adverse cellular and tissue microenvironment. So look at it like the drain is starting to get clogged, the sludge is building up, and you're starting to see some dysfunction. At almost its latest stage, in advanced stage, we see widespread senescent cells causing inflammation. It reaches critical levels. You get all kinds of diseases related to that, like cognitive decline, cataracts, etc. The drain is clearly clogged. The water is overflowing, and the bathroom is an entire mess. And so in senotherapeutics, you you give an episodic senolytic therapy, which cleans out or removes these senescent cells over a defined therapeutic time period. You remove them back to the time when there were few senescent cells again. It allows the organ functions to return to an efficient manner. You can monitor the level of senescent cells in the body over time. And when they accumulate it again, you just can retreat. So you don't necessarily need to treat continuously. You can have an episodic therapy, which can reduce the risk potentially of side effects. So, as I've said, it's becoming more and more clear that the accumulation of senescent cells in the body over the lifetime, I think there's no controversy that it is, a contributor to aging, but in our opinion, it is the primary driver of aging, including all the problems associated with aging, as I've mentioned. And that there's clear, compelling proof of concept in animal data that eliminating those senescent cells has beneficial effects in reducing, eliminating all of these diseases with a single gerotherapeutic approach. Now, at the recently held ARDD meeting, which is a meeting of all the leading aging researchers in the world that was held in Copenhagen in August, one of the leaders in the field basically said that our greatest hope in developing these types of medicines is to see what's already approved and try and repurpose them. So I feel that's a generally pessimistic view of where the current state of the field is. Now, I've been working on this since a little over a year ago when I did this study, which I call Self-15, where we took naturally aged 75-week-old mice, which is approximately equivalent to a 50 or 55-year-old human, and we treated them over 8 weeks with some peptides that we had identified that appeared to be effective senolytics. And then we monitored their tissues. We sacrificed them and looked in the tissues to see the number and extent of senescent cells in the body. Now, since then, we've done a fair number of additional studies. We're up to cell 27 here now, where we've added another peptide, 2107 to 2101 and 2105, which are our senolytic peptides. We do extensive phenotyping of these animals and evaluate them. And this is an ongoing study, for example, where we're treating these aged mice with these peptides and then evaluating them in a number of ways. We have a room at our facility that runs 7 days a week in phenotyping these mice with a number of instruments in the room. We have a motorized treadmill, a rotor rod, a maze, a grip strength, a DEXA scanner, all evaluating these mice that we're treating with our senolytic peptides. This, for example, is a rotor rod. It's all automated, so you put the mice on and you get the data readout, which is free of any observer bias. Now, I submitted an abstract with some of our data to the International Conference of Frailty and Sarcopenia, which is going to be held in March of 2025 in Toulouse, France. The abstract's titled "Novel Senolytic Peptides Alleviate Sarcopenia and Frailty in Naturally Aged Mice by Targeting Senescent Cells." And I plan to unveil some really remarkable data at that meeting. But because it's an academic meeting, I'm trying not to reveal the results of our data until then. But I will give you a little bit of a sneak preview, or if somebody's interested in this more and wants to go under CDA, we could show more. But in summary, in the two studies I just showed you, cell 15 and cell 27, I think we have clear evidence that we've discovered the first gerotherapeutics that are aimed at an aging-specific target. Now, what do I mean by that? So, first of all, these studies were all performed by my talented team at Eos SENOLYTIX in Houston. They're all performed in naturally aged 75-week-old mice, so it's not a transgenic model or some other artificial model. And as I've said, a 75-week-old mouse is roughly the equivalent of a 51-year-old human. We have three peptides that we've tested extensively. Two of them we're planning on developing clinically. That's 2105, 2107. There are proprietary senolytic peptides. They target senescent cells for elimination with about 8 weeks of treatment. So we've shown now, and I've shown you data, that senescent cells play a widely recognized role in aging. And if you target them, you're avoiding targets that play a diverse and key role in normal cell biology. So we're not targeting mTOR or BCLX or even the Yamanaka factors, which I applaud Daniel on his work there. But as he noted, there's risk that he's trying to mitigate by improving them. We're just targeting senescent cells, which play a very limited role in healthy biology. And our therapies only require episodic therapy, so not continuous. And in summary, what I'm not showing primary data except for one sneak preview, but in summary, what we've established now and what I'll be presenting in March at that meeting is that our peptides effectively and safely eliminate senescent cells. More than 75% of senescent cells in an organ can be eliminated within 8 weeks of therapy. That we also show a reduction in the plasma circulating levels of cytokine components of the SASP in response to removing these senescent cells. We show tissue morphometry improvements like the intermyocyte space in muscle, the adipocyte area in treated mice, all improved. We show prevention of weight loss that's typically seen with age-associated sarcopenia. And remarkably, we show preservation of lean body and bone mass at the expense of fat mass in these animals. We show improved performance on all of the apparatus I mentioned, the motorized treadmill, the four-lever grip test, the water-motivated stone T-maze, and the rotor-rod. Now, I told you I'd give you a sneak preview. So here's just one piece of data from cell 27 data. These are 75-week-old mice that were treated with these three peptides for 13 weeks, and then we did DEXA scanning on them. And I drew a line here through the lean body mass percent of the negative control saline mice, because it's a little hard to see the data, but you can see that remarkably, after 13 weeks of peptide treatment with the two peptides that we're developing clinically, 2105 and 2107, we see a 20% reduction in fat mass and almost a 10% increase in lean body mass, including an increase in bone mass. So this is important. I don't know if you saw data recently from the Amgen compound in clinical trials where there was some concern over loss of bone mass in the high dose group. Here we're seeing preservation of bone mass and lean body mass at the expense of fat mass. Now, you might ask, was I surprised to see these results? Well, I really wasn't surprised. And why not? Because in 2017, Farr et al. Already had shown that if you eliminate senescent cells in this genomic ablation model using the INK-ATTAC suicide transgene, if you eliminate senescent cells in aged mice, you would see improvement in the bones. So I already knew that if you could just develop a therapeutic that could do the same thing, you could see these results. Now, as a very quick side note, the data that was generated here using the INK-ATTAC model was developed using technology by Baker et al. That actually my group had invented in the past that we used to launch Bellicum Pharmaceuticals, which was one of my first companies, one of the first CAR T-cell companies that we took public in 2014. We had invented caspicide, which was a cell therapy safety switch. It's the only validated switch in humans. And that's what Baker et al. And Kirkland used for their seminal papers, and everybody else who used that model to show that elimination of senescent cells leads to beneficial effects. But that's actually our technology that he was using. And so we're actually developing it now as a gene therapy in a separate company. So that's how I got into the field. So just to finish up here, I think senescent cells are the culprit in a lot of aging-related problems, but few drug developers are pursuing what I think is a clear target. I think mostly because it's been very difficult to find something that could selectively eliminate senescent cells from the body, both safely and effectively. But we've discovered a number of senolytic peptides with the remarkable ability to effectively and dramatically eliminate senescent cells throughout the body, including in the brain, with no apparent side effects now out to 14 weeks of therapy. And so we're going to take this forward and try and jumpstart the entire field of xenotherapeutics. by performing 3 phase 2 clinical trials simultaneously with 3 different primary endpoints using 2 different peptides, the 2107 for whole body, 2105 for brain. We're targeting obesity and glucose metabolism because we've noted that we can preserve lean body mass at the expense of fat mass, sarcopenia and frailty, and cognitive disorders. All of them have different primary endpoints, but we're synchronizing the secondary endpoints across all of them, similar to what Daniel was saying, to try and get a broader scope of a gerotherapeutic claim as opposed to individual diseases. We're currently raising 100 million Series A to do this ambitious program, and we're hoping to close it by the end of the second quarter. So 100 million targeted Series A. We've raised about now close to 10 million convertible notes. We have a number of peptides that we're taking forward. And if you're interested in participating in this institutional round, you should contact our investment banker, John Andreas, Evolution Venture Partners. And then just finishing up here, I'm the founder, chairman, and president of the company. I founded Bellicum Pharmaceuticals, which is the first CAR T-cell company, took it public in the past. And I've been a very active researcher in my previous career. So this is really a science-based company. Jody Aiken is joining us, who was the founder of Hawthorne Health and who got the TAVR approved for Edwards Life Sciences, and she's really great. And Kayvon Nambar, who's a very well-known person in our field. So that's all I have. I'm happy to talk to anybody who communicates with me if they're interested in this round and go under CDA. I'd be happy to share our really remarkable data prior to the March meeting. But in March, we'll be presenting a very extensive set of data that I think is going to be groundbreaking. Thank you very much.
Kevin, thank you. Well, obviously, there's a lot to unpack there, as you say, very ambitious. So I'm glad you're going to be joining us for the panel session at the end. So I'll keep my powder dry on some of those questions. And thanks for your presentation today. Really good.